Welcome, everybody. Welcome, everybody, to the 2023 Hans Fischer Lectureship. I am Josh Miller, Professor and Chair of the Department of Nutritional Sciences, and I'm so happy to see you all here today. It's a beautiful day outside. We're gathered to hear what will undoubtedly be an inspired and thought-provoking seminar, and we're not on strike anymore. What could be better? Before I say a few words about the lectureship and introduce our lectureship namesake and introduce our guest speaker, let me first hand the podium over to Executive Dean of the School of Environmental and Biological Sciences, Laura Lawson, who will have a few words of welcome.
For those of you who have lectured in this room, you know that this podium is like when you have someone working on it, you just don't go anywhere near it. You kind of just stay away and let the magic happen. Welcome. Happy Earth Day, one day early. It's very exciting to be here today and on such a beautiful day. Hopefully later they can open up the back for you to just enjoy this beautiful spring day. It does definitely feel like we've turned over into a new season, and it's absolutely delightful to be here. So it is my pleasure also to welcome you to the 2023 Hans Fischer Lecture. Really, this is an essential moment in our school's academic year. It's part of our history and our commitment to being a really unique place of learning and connection. And I know you're going to be hearing from Hans in just a minute. It's also really wonderful that this is a lectureship named after someone who devoted 52 years as a faculty member in various roles and really shaped Rutgers as it is today. So it's an honor to have this, and it's exciting that he can attend as well. So hello in Zoom land to Hans. Providing a welcome to this lecture is a special treat also because it is an opportunity to be part of the nutritional sciences community, joined together in an annual tradition around a shared interest in learning new things. And as I reviewed the list of past lectures, it's like a departmental diary, a glimpse into your discourse as a community. And today, as we gather to hear from Dr. Stover about enhancing the purpose of food, I look forward to hearing, learning things and also hearing the discussions that you will have. And while I'm here to welcome our guests and welcome this event, I also want to take this opportunity to thank the Department of Nutritional Sciences, the faculty, staff, students who make up that group, and alumni, and all your partners who make up the Department of Nutritional Sciences. Life is really demanding right now, and there's so much going on in the world that for you to take this time and this commitment to running an event like this, that says a lot. You know, these kinds of events start getting planned, if not yesterday, definitely this afternoon, right? And it takes a lot to make these things happen and to bring everyone together with the strike, with the semester ending, with just all of the things that are going on in life. It means a lot to bring you together, and I am really appreciative of everything that you're doing to keep this part of our important mission alive. So thank you to the department. And so with that, I will hand this back to Josh.
Thank you so much, Dean Lawson, and very much appreciate you being here today. The Fischer Lectureship honors the legacy of the founding chair of the Department of Nutrition in 1966, Dr. Hans Fischer. Dr. Fischer was hired as an assistant professor of nutritional biochemistry at Rutgers in 1954. His research in various animal models, including chicks, rabbits, and humans, provided fundamental insights into the roles of amino acids, vitamins, and fiber in metabolism, egg production, growth, wound healing, stress management, neurotransmitters, and behavior in alcoholic addiction and withdrawal, and cholesterol in atherosclerosis. It is upon this legacy and his many contributions to the Department of Nutritional Sciences and Rutgers University that we hold the Hans Fischer Lectureship. And it is my honor to let you know that Dr. Fischer is attending today via Zoom from his home in Boston. Please join me in acknowledging Dr. Fischer. And I would ask, Hans, did you want to say a few words from the, what I call the God mic in the room?
Yes, I would be very happy to do so.
Okay, go ahead.
Just to give everyone an opportunity to understand how this lectureship came about. In 1991, 32 years ago, a small committee was asked to review the Department of Nutritional Sciences, which, as you heard, got started in 1966. And the chairman of that small committee was Dr. John Suttie from the University of Wisconsin, who was an expert on vitamin K. And he recommended, after giving the department very high marks for its pedagogical and research undertakings through all these years, recommended that it would be very appropriate to add a lecture which would bring outstanding scholars to Rutgers and to give the faculty, the students, postdocs an opportunity to hear such outstanding people once a year, and at the same time also to honor me for my role at Rutgers University. And so that's why this lectureship came into being 32 years ago, and we've had many outstanding people, including some of my children who are researchers, and of course today, Dr. Patrick Stover. And so without any further ado, I turn this back to you so we can continue on to the lecture.
All right, thank you, Dr. Fisher. Okay, before moving on to introducing our distinguished guest speaker, first let me acknowledge Christina Duffy, who every year makes this event happen in our administrative roles. And I want to also thank our seminar committee chair, Harini Sampath, and members Malcolm Watford, Sue Shapsis, and Joe Dixon. And now to today's speaker. Dr. Patrick Stover is director of the Institute for Advancing Health Through Agriculture at Texas A&M University. Previously, he served as vice chancellor and dean for agriculture and life sciences in the Texas A&M AgriLife program. And before that was director of the Division of Nutritional Sciences at Cornell University from 2005 to 2018. Dr. Stover is well known to most of us, and his list of accomplishments and awards is extensive. Here is just a partial list. In 1997, the Presidential Early Career Award for Scientists and Engineers, awarded by President Clinton. In 1999, the E.L.R. Stockstead Award in Nutritional Biochemistry from the American Society for Nutrition. In 2014, also from the Society, the Osborne and Mendel Award. 2014, Fellow of the American Association for Advancement of Science. 2015 to 2016, President of the American Society for Nutrition. 2016, elected member of the National Academy of Sciences. Current editor for the most highly cited nutrition journal, Annal Reviews of Nutrition. And just very, very recently was named as the U.S. Department of Agriculture Agricultural Research Service W.O. Atwater Memorial Lecturer. Now, on a personal note, having Dr. Stover with us today is very meaningful. In the field of folate and 1-carbon metabolism, he has made major contributions. To highlight just one, Dr. Stover is responsible for working out the details of nuclear folate metabolism and how key folate and 1-carbon metabolism enzymes are transported from the cytoplasm to the nucleus via a process called summoylation. I only know what that word means because of the work of Dr. Stover and his team. Over the years, I was privileged to see this story unfold as he revealed their progress in working out the details of nuclear folate metabolism at national and international meetings through clear and engaging presentations of elegantly designed, masterful experiments. Now, before I hand the podium over to Dr. Stover, in preparing for today, I wanted to see if there were any connections between him and Dr. Fisher. And so I looked through the literature, and here's one of Dr. Stover's papers. And I want to point out that he was working as a postdoc with Barry Shane, who many of us know as a longtime researcher in the area at the University of Berkeley. Now, Barry Shane worked with E.L.R. Stockstead, and one of the awards that has been awarded to Dr. Stover. And I went back to Stockstead's first paper that I could find online, and it was looking at trying to figure out dietary water-soluble factors required in chicks. And what I can tell you is that there weren't any specific papers published between Dr. Fisher and Dr. Stockstead, but the work that Dr. Fisher did early on in his career actually used the same model of chicks. So, put that all together, 3 degrees of separation between Dr. Stover and Dr. Fisher. And with that, I pass this over to Dr. Stover.
Thank you very much, Josh.
I think you'll need to use the mouse, yeah.
Well, thank you very much, Josh. So I didn't quite know what to do with this presentation because I'm a metabolic biochemist by training. If I talk about folate for an hour, you're all going to fall asleep. And I knew some of you were interested in what we're doing at A&M in terms of the institute we're building. So I attempted to put those two together, and it makes sense in my mind. We'll see if it makes sense in your mind. Well, part of this is because, and it was something that Bert Garza, who was the guy, the director at Cornell who hired me, who from the very first year I was there as an assistant professor, he says, don't be an idiot savant. He says, do your lab, do all that stuff, but you have to do service as a land-grant university to national and international organizations so that we bring the very best science to these other organizations that are involved in policy and so forth. So what I'm going to talk about today is going to mix both of those. It's informed both by my own research, but also by work over the years related to folate and fortification with the World Health Organization, with the CDC, and national academies, and other groups. Because a lot of that really is what translated into what we're trying to do at A&M with the Institute. So we'll see if this makes sense or not. It may not, but we're going to try. But enhancing the purpose of food is very intentional. You're seeing right now at USDA and NIH, there's all of these initiatives now in precision nutrition. You're seeing them now in food is medicine or food as medicine and all this other stuff. But what this really is about is enhancing the purpose of food. And so, why do we have the food system we have today? That's a fundamental question. Why do we have that? The food system is the basis for, obviously, all of the nutrition that we have. And the food system we have today is intentional. It came out of the post-World War II era, where there was a lot of hunger coming out of the Depression through the war in the United States. And there was an intentional decision made that hunger was unacceptable. It was a moral imperative that not only in the US, but around the world, we had to eliminate hunger. And because of that, we scaled an agricultural system to make hunger rare. And we did it by producing calories. A simple calculation. How many people in the world are there? How many calories do we need? We scale agriculture towards that goal, and it was unbelievably successful. Since then, there's still hunger, but it's not because of a lack of food production. It's because of access issues and others, but that's why we have the food system we have today. That's why we have the economic model around food systems to make it cheap and affordable, low margins for farmers, but low cost than food costs less here in terms of percentage of income than any other country, et cetera, et cetera. It was very intentional, and the purpose of the food system was to eliminate hunger. Now, at the same time during World War II, there was a lot of efforts to learn about food and soldier performance and such and so forth and so on. But that wasn't the purpose of the food system. The purpose of the food system was to eliminate hunger. And if you look now, we still have to produce more food. The world population is growing. There's going to be another 2 billion people on this planet. You can see there where they're going to be. It's India, China, Nigeria, and the United States. So we need to still produce more food and more calories and more protein to feed people. But at the same time, we're seeing another problem. And that is, you're seeing that right here, that the food system we have today makes people sick. Diet-related chronic disease is the biggest driver of healthcare costs. Estimated somewhere between $4 and $6 trillion a year to the U.S. economy. Nobody can afford those costs. What do we do about that? So we still have to grow more, but we have to do better. And of course, you've seen these maps. This is a fairly recent phenomenon, and it continues to accelerate. There you see the prevalence of type 2 diabetes from 2004 to 2019. This next slide is heavy, so it's going to take a second to show up. But you also see the obesity prevalence maps. But what you also can see from here is that the prevalence of obesity is not evenly distributed across the U.S. population. It is especially enriched in our underserved populations. So if you want to lower healthcare costs and lower diet-related chronic disease, because we all pay the healthcare costs, that's where you have to focus, because that's where the major problem is. And we also see now effects on life expectancy for the first time, where life expectancy was increasing for decades. Now you're seeing, since the pandemic, decreases in life expectancy. And again, you're seeing it's not evenly distributed. You look at non-Hispanic, white, and Asians and compare that to some of the Native American and Hispanic populations. So you now have this disparity as well in life expectancy that's continuing to grow. And we saw during the pandemic a lot of the social unrest. This isn't sustainable. You can't have a functional society where you have this sort of disparity. And it's, again, especially acute in this country, and a lot of it is rooted in food, in the food system we have today. This was recognized by Congress in 2020 in the Agriculture Appropriations Bill. There is specific language that the USDA shall report back to Congress within 180 days how agriculture and food can be used to lower healthcare costs. Nobody knows how to deal with healthcare costs. Food's the major driver. Food has to be the solution. So how do we deal with this? So if you look, again, back after World War II, this was the purpose of the food system. The purpose of the food system was to produce and overproduce in abundance food, fiber, and fuel for the economy and to eliminate hunger. Now you see the emphasis on food for health. What does that mean? You see a lot about the environmental footprint of agriculture. What does that mean? And we have to make sure that all of this is economically viable, because if the economics don't work, nothing's going to work, right? And just a word about the environment. Not only does agriculture have an environmental footprint, I'm not going to be talking about that, but what we're seeing is challenges with the environment and our ability to produce food. So the environment is changing. And the problem is it's changing in variability as well. It's more stochastic. And this is a real problem. If things get hotter, we can breed for hot. If things get colder, we can breed for cold, right? Well, what we're getting is extreme variations in hot and cold and wet and dry in the same season. We can't deal with that, and it's beginning to affect yields. It's beginning to affect our ability to produce food. So climate variability is a huge issue. We also see, again, healthcare costs. And what do we do about that? And remember, the food system we have today, the economic model is perfect for its intent. The intent is to produce food that can be affordable and accessible by everybody. That's the economic model. Well, what it does is it creates externalities, and healthcare costs and environmental costs. And so you see some people who want to internalize the externality by taxing soda or this or that, which is, most people will tell you, really bad economics. An economic model has to be geared towards the endpoint. If the endpoint is hunger reduction, that's the economic model we have. If it's health, we need a different economic model. But nobody knows what that is yet. They're trying to internalize the externalities like you see in true costing and these other things are actually bad economics, a lot of economists will tell you. And we also see, because of the economic model of keeping margins really low for farmers, right? So the food is affordable. Well, farmers are selling their land to development, to solar farms, because it's more profitable for them. And so the point is, the whole system's in a little bit of trouble, right? It's not making economic sense, it's not making environmental sense, and it's the healthcare costs and, having high rates of chronic disease are problematic. And so the question is, not this nature, can science feed the world as the population grows, but can we feed the world in a way that promotes health and is sustainable? And so the biggest challenge we see is you have these really four independent, complex dynamic systems. You have food, you have people and their health, you have the environment, and you have the economics. And again, they were all aligned around the purpose of reducing hunger. All of those have to be adjusted if we're going to change the endpoint to promote human health. And how are we going to do that? And so the way a lot of this started was back in 2017. The National Academies was asked by Congress to change the way that we do dietary reference intakes. I think you all know what the dietary reference intakes is in that process. And we used to set them for hunger, if you will, hunger or hidden hunger. That is, how do you maintain adequacy? How do you get enough of all the vitamins and nutrients and such so that you have sufficient amounts for function? And Congress said, well, we don't have scurvy anymore. The pirates are all gone and this and that. The problem we have is chronic disease, so change the outcome from hunger to health. And so I was part of this committee that began to look at how we develop a framework for changing the endpoint of RDAs, which is essentially the food system, from hunger to health. And of course, this is really, really challenging. Because number one, hunger, or lack of an essential nutrient, hidden hunger, is a one-to-one correlation. There's one cause, generally, if you're healthy, it's you're not getting enough of it. And so most of the population responds in a similar way. If you use a health outcome, it's much more complex. Because number one, diet's not the only driver of chronic disease. It's a combination, right, of your stress, your sleep, your exercise, et cetera, et cetera. And so we have to think completely different about what does food mean, what do nutrients mean in terms of chronic disease reduction. We also have to think more in systems because health or disease is not related to one nutrient. When you talk about a chronic disease, it has to do with network failure, it has to do with accelerated aging, these sorts of things. So we have to think very differently. It also, which people don't appreciate, is there are a lot of non-essential bioactive food components that promote health. Now, they're not essential, so they never had a DRI before, but now they can have a DRI. If you can show an individual nutrient reduces risk of chronic disease, it then will get a DRI. It'll then feed into the dietary guidelines for Americans, goes into all food assistance programs, so it's a big opportunity for producers if there are food components in what they grow, if they have a health effect. And people will begin to take advantage of that. And the other issue, as Francis Collins says, is responders and non-responders. People react differently to food in the diet-disease relationship. Not everybody responds the same. There's heterogeneity. This is what's brought in the precision nutrition initiatives, is how do we classify people based on how they're going to respond I like to think of it this way because I do a lot of mouse genetics. Penetrance, what percentage of the population is going to show an effect? And expressivity, how bad is the phenotype or how good is the phenotype going to be resulting from that food exposure? And so I'll be talking a lot about penetrance and expressivity. And so again, that is the biological premise of precision nutrition. It's well supported by evolutionary biology. When we look at genomes, human genomes, we look at variation within human genomes and what genes are evolving quickly, which genes haven't evolved much at all. It's those related to food and nutrition that have evolved the fastest, giving us the most variation we have in the population, that and immunity. Because as populations emerged all over the world, they had to adapt to local food systems. If they didn't adapt, they didn't thrive, they didn't expand. And so that's why you see two of the greatest selective pressures in genetic variation we have today in the population is due to, again, food and immunity. Just want to highlight this study, which I think was great. David Threadgill, my colleague at A&M, did a few years ago. But just quickly, he did a study where he took 4 different mouse strains, inbred mouse strains, fed them 4 different diets, aged them out, and asked the question, what's the best diet? And he measured everything, collaborated with everybody, epigenetic decay, he looked at mutation rates, he looked at biomarkers of everything, he looked at disease, he looked at this. You couldn't say that one diet was better than another. It depended what mouse strain and what outcome you were interested in. That's the challenge we have, and it models really well in mice. So how do we begin to think about this? Because we know one size does not fit all, as Francis Collins says, in the diet-disease relationship. So how are we going to get there? What's the pathway going to be? And what I'd like to share with you was a little bit of the folic acid story, folic acid and neural tube defects. Because in that National Academy report, I showed you the Chronic Disease Framework Report. They use folic acid as an example, birth defect prevention, of the things we have to think about in terms of using food for health outcomes rather than functional or adequacy outcomes. So this was a huge success. There is no success story in nutrition like this. Well, what we knew is that there is something called neural tube defects, a birth defect that happens very, very early in pregnancy, often before a woman even knows she's pregnant, when you have closure of the neural tube. It's one of the fastest times of cell proliferation in your entire life where the neural epithelium has to expand and close. If during this critical window it does not reach closure, the neural tube will remain open throughout the life of that individual, and that individual will be paralyzed from the site of the lesion down. If it doesn't close in the head, that's going to be lethal. So these are extremely debilitating birth defects. And what we know it has a genetic component because it runs in families. We know it runs in families, but we also know that there was a nutrition component with this as well, and we'll talk about that. But the important thing I want you to know is it's about 2,500 affected births a year. Small number, big effect. High healthcare costs, big effect. But it was known that low folate status in predisposed pregnancies. Most women, if they become deficient in folate, are not at risk, but some people are. Who are they? We don't know. That's the challenge. Well, what we know back in the 60s, it was noticed that women who presented with a child with a neural tube defect tended to show biomarkers of impaired folate metabolism. This later then went to clinical trials in the early 90s, and in fact it worked. When you gave women supplemental folic acid, you were able to reduce risk as much as 70%. That then went to public health recommendations. If you're going to get pregnant, have folic acid. Well, most pregnancies in this country are unplanned, so that didn't work, right? So that led to a fortification of the U.S. food supply with folic acid for the purpose of reducing the spina bifida, this birth defect. So what's interesting about this is the first time we used food policy, not because there was a deficiency in the population of that vitamin. There was not. There was no folate deficiency to warrant fortification. Every other fortification initiative was done because of that. This was done for a medical purpose, using food as medicine, using fortification to help 2,500 births every year and to prevent that birth defect. And everywhere where fortification has happened, it's been successful. And if you look here, these are countries that are fortifying right now their food supply with folate. This is actually an old picture, but it's okay. And if it's dark blue, it's mandatory. If it's light blue, it's voluntary. But you see some white there, especially in Europe. They don't fortify. So why do you have something that's cheap, it's pennies, to put folic acid in the food supply? It works everywhere you do it, but some countries don't do it. Why is that? And that's, again, because of a lot of nuances. Number one, it was the first fortification that should have been there. Not meant to remedy a nutritional deficiency, it wasn't there. It was to prevent this birth defect. Number two, it exposes everyone for 2,500 births, right? And so some have asked, well, and it's hypothetical, well, maybe there's another small group that accrues risk for this, right? But that's public health. Public health assumes benefit risk analysis. It's not do no harm. But that's a question people ask. We don't know the mechanism. We have no idea how folic acid prevents birth defects. All we know is we have a black box where you give folic acid in a clinical trial or in the food and you prevent NTDs. It works, but we don't know what this connection is, and there's others concerned that it may be increasing cancer risk for other people. Again, there's no strong evidence for it, but it's been raised, and you can make, a hypothetical argument for that. There's other observational studies that raise concerns about not all of it getting metabolized, but all of this exacerbates common concerns of unintended consequences. So think about trying to use a food supply to lower rates of chronic disease. You're going to run into the same thing because people respond differently to food. And so this idea of how do you take a food supply that everybody is exposed to and manage risk-benefit, or be able to identify and classify people based on precision for what diet is best for them. So it just illustrates part of the issue. What we have been doing in our lab is trying to understand what this mechanism is. Because if you understand how something works at a molecular level, then you can begin to find biomarkers and you begin to classify people and then target in a way with precision those who need the intervention or those that need whatever the dietary exposure they need to prevent the chronic disease. So Josh showed this. This is one carbon metabolism. I don't want to go into this too much, just to say that folate is a B vitamin, and it just carries and activates one carbons like formate and puts them into purines for purine biosynthesis to make thymidylate and to remethylate homocysteine to methionine for methylation reactions. So it's a network, it's interconnected, but you just get these three outputs, right? It is a tightly integrated network. If you tickle this network over here, you'll see repercussions over here. So it is tightly, there's a lot of crosstalk between these pathways. Well, what we found that during S phase, and there's a number of wonderful students who did this, during S phase, this pathway gets this SUMO tag. SUMO is just a polypeptide, a cousin of ubiquitin, and it goes into the nucleus. It forms a metabolic complex right at sites of DNA synthesis. So that T-base is made on-site, on demand, at the replication fork during DNA synthesis and repair. So it is the only base of DNA that is made on-site, on demand. And this is unique to higher mammals. Yeast don't do this. Yeast leave it in the cytoplasm. Only higher eukaryotes put this pathway into the nucleus. And we were really interested why that is. And we also began to knock out some of these enzymes, so forth and so on. And when we did, guess what? We got neural tube defects. When we knock out SHNT1, we just need to knock out one copy, not both. We get about 10% penetrant neural tube defects. You see there, the head never closed. So that led us to believe then that this pathway in the nucleus, that takes tetrahydrofolate to make 5,10-methylene tetrahydrofolate to give that activated one carbon to go to DU1P to make thymidylate that then goes into DNA. Somewhere in here, you have risk of a neural tube defect if you disrupt it. How does it happen? Well, if you don't have enough of the DT base, cells grow slower. That's known. Thymidine can be rate limiting in cell growth, less proliferation. We also know that if you don't make DT, DUMP will accumulate, it will become triphosphorylated, and it will get misincorporated into DNA. So not only do you get slower proliferation, you begin to get uracil in DNA. And what's the uracil doing in DNA? Well, it's repaired, but does it have other types of functions as well? But somewhere in this pathway is, we believe, the etiology of neural tube defects. And we really like this mouse model because there are about 200 mouse models that give you neural tube defects. But if you knock out any gene that affects proliferation, you'll get a neural tube defect. But these mimic or phenocopy the human NTDs. There's a variant in SHMT1 that inhibits nuclear input. It doesn't totally inhibit it, but it decreases it. It's a risk factor for neural tube defects. If you look here, you just need to knock out one or both copies, we see neural tube defects. The majority of NTDs in humans are folate responsive. Ours are folic acid responsive. If these mice are folate replete, we don't get neural tube defects. We have to make them folate deficient. If we take a normal mouse, a wild-type mouse, and make it folate deficient, it won't show a neural tube defect. No animal will. They have to be sensitized. This gene sensitizes them. Low invariable penetrance for both, and a subtle alteration of metabolism. When we look at these mice, and we look at all the biomarkers in serum that are related to folate and this and that, none of them are different. There is no serum indicator for neural tube defect risk or impaired folate metabolism in these mice. So we think it's a great model. So I just want to review what I just said here, is that at sites of DNA replication, we've shown that there's a multi-enzyme complex that makes thymidylate. What I didn't tell you was SHMT1 is the anchor. It's the scaffold. It binds lamin like a rock at sites of DNA synthesis. These enzymes all get sumoylated. The sumo is the Velcro that holds them all together. And you get this metabolic complex then with SHMT serving both as a catalyst supplying 1 carbon units, but it's redundant with this enzyme that is also supplying 1 carbon units. This is formate, this is serine, different sources, but they're all together here to make thymidylate. If we knock out SHMT1, this complex breaks up and these enzymes are just all floating around in the nucleus, not in a complex. We've been able to show all that. So in order to get a mechanism, we want to ask the question, what are the elements of SHMT1 that causes NTDs? Is it its catalytic activity? Is its ability to bind folate? And that's important, because when you have enzymes in a metabolic complex, it's been shown that that folate is channeled from enzyme to enzyme. It's not free in solution. It's actually transferred from active site to active site. So we wanted to say, well, is it a channeling effect, folate binding activity, or is it the scaffold activity? That's important. And so what we did was make the following enzymes, or mutations in enzymes. Here we have a wild-type SHMT1. This is from the crystal structure. Here's folate. It has a folate binding site. This tyrosine here is critical for folate binding because you get resonance overlap between this tyrosine and the paraminyl benzyl glutamate of tetrahydrofolate, essentially a stacking interaction here that really binds, or rather drives the binding affinity. If you look here at the active site, you have vitamin B6 here, pyridoxal phosphate. These two residues here are really important for catalytic activity. So we took one mouse, we did site-directed immunogenesis, and we knocked out its ability to bind pyridoxal phosphate, yet the enzyme's intact, it folds, can do everything, it's just not a good catalyst. We took another one where we kept this disrupted catalytic site, but then we also knocked out this tyrosine so that it can't bind folate either. These enzymes express in mice just fine. They form the metabolic complex, as you can see. I'm sorry, I'll just review here what we have. The mice we have a wild-type mouse that has all the activities, a knocked-out mouse that has no protein, and then we have two mice. Both don't have catalytic activity. One can bind folate, one can't. And again, they express just fine at mice at the appropriate levels. You can do pull downs, IP, they're still on the complex. They are perfectly fine proteins, they just either can't catalyze reactions, or they can't bind folate. So we took our mice here. This is our knockout mice, put them on a folate-deficient diet. This one here has no activity and it can't bind folate. This one can bind folate, but no catalytic activity. We keep the mice, the female mice, folate deficient. We mate them and we look for neural tube defects and look at what we find. This is the knockout mouse. If you look at the number of NTDs, it's about 10%. That's what we always see. If we look at the enzyme that's intact, but can't use serine as a 1-carbon source, no activity, same effect. So if you knock out the activity, you get the same thing as knocking out the protein. But if you now knock out the activity and the ability to bind folate, resorptions double, NTDs go up by 30%, and severity of NTDs, I'll show you what that means in a minute, skyrocket. So having a protein that can't bind folate is worse than not having the protein at all. This is what I mean by severity of NTDs. These are unaffected mice. This is a simple exencephaly. This is what we call craniorachescesias. The head didn't close, but neither did the spinal cord. The lesion goes all the way down the spinal cord. That's a catastrophic neural tube defect, and those are more prevalent. So what does this mean? What this means is that if you knock out SHMT, these enzymes are still floating around, but you can make sufficient thymidylate to get mice. Some of them will have neural tube defects. If you put an inactive protein in here, you get the same effect. But if you put one that can't bind folate, the phenotype is worse in both penetrance and severity, telling us that folate is channeled here. And if you impair the ability of this enzyme to bind folate, you essentially constipate the whole system, and it's worse than not having any protein at all. So why is that important? It's important now that we're beginning to understand mechanism at a higher level, and hopefully being able to find some marker again that we can find out in humans and be able to identify humans where the defect is in terms of metabolism that's leading to the neural tube defect. Second question we wanted to ask is, does uracil accumulation impact NTD risk? And so if you don't have enough T-base, you're going to incorporate a U. The U is going to be on the DNA strand. There's going to be an enzyme, uracil DNA glycosylase, that's going to excise that uracil, causing an A-basic site, and then you're going to get cleavage of the DNA backbone and then repair. So this is what happens when you misincorporate uracil in DNA, and that's the only molecular phenotype we see with this mice is they have more uracil in DNA, they have more repair. So we want to ask the question, well, is it the uracil in DNA that's causing the neural tube defects, or is it the repair of the uracil that's causing neural tube defects? So we took mice that were UNG negative, they can't repair DNA. We took our mice that don't have SHMT, they incorporate uracil into DNA, and then we crossed these so that these mice are going to accumulate uracil in DNA and not being able to repair it. And these are the data. Again, if we look at SHMT null mice, 11% have neural tube defects. If we look at those that can't repair UNG1, no neural tube defects. If we cross these two, we lower the incidence of neural tube defects by about 60 to 70%. So this tells us that it's the DNA repair that's causing the neural tube defects. It's not a proliferative effect. It's effect of uracil being misincorporated into DNA because you don't have enough of the enzyme, that uracil is repaired, and it is the act of repairing that leads sometimes to apoptosis and other effects that is the source of the neural tube defects. So neural tube defects are due to effects of hyperactive DNA repair. And that's essentially what that says. So how does this relate to the challenge of precision nutrition? What the folic acid story illustrates is, number one, there's going to be benefit and risk, like we saw with fortification. Some people are benefiting, but others may be accruing risk. And so how do you tailor an entire agriculture and food system when there's a heterogeneous population and people are going to respond differently when you have that health outcome? How do we deal with the issue of population versus precision? What does that even mean for dietary guidelines if people react different to food in terms of a disease outcome, which we know they do? What are the evidentiary standards we're going to use for benefit and risk? With normal tube defects, we see from the clinical trials there's definite benefit. There's no real evidence, quote unquote, statistical evidence for risk, but how do you deal with that uncertainty? And how's the public going to react? We need biomarkers. We need to understand and be able to get that to an individual, what their risk is and how food can mitigate that risk. And we do not have the biomarkers to do that, but we need that both to identify and classify people who respond to different dietary exposures so that they have the agency to make the right choices. Issues of penetrance, of expressivity in phenotype that vary among people. And of course, the public trust, because right now we don't do really good with public trust in any Pew survey as nutrition science researchers. We have to get this right with this precision nutrition, or nobody's going to listen to us. So lots of issues there that we have to solve if we're going to move forward both precision nutrition and food as medicine. But on the other hand, we have the tremendous capacity to do whatever we want to the food system. We can use CRISPR, we can change pathways, we can put higher concentrations of metabolites or vitamins or nutrients in, take them out. We can do whatever we want to do if we can get over the barriers. It's not the technology that's the barriers, it's the regulation and the acceptance of those barriers and knowing what we want. Because we still have to know what do we want out of the food system that's going to give us those health outcomes. So what we've done at A&M, and some of this came from support from the state with an $18 million recurring appropriation and then match that with federal funds and other funds, to set up an institute that we call the Institute for Advancing Health Through Agriculture. Where we said if we're going to solve these problems, we have to deal with the agriculture food system as a system. You can't do nutrition over here and soil science over here and so forth. It all has to work together, and we need to take every opportunity we can to achieve that outcome of human health. And so we developed audiences that we were interested in, the production side of the ag chain, consumers, put this under the umbrella of the Institute for Advancing Health Through Agriculture. It actually had a terrible name. And I was talking to a New York Times reporter who said, that's a terrible name. This is the name you should use. So that was totally stolen from the New York Times. We also, the other third important audience that we were interested in, and that is decision makers. That is policymakers. If you look right now, decisions around the food supply aren't really driven by science. They're driven by preferences, values, and beliefs. And it's a big problem. And so what we have done, copying off of what are very common in the medical community, is an evidence center. We have created the first agriculture, food, and nutrition scientific evidence center. So if any person, decision maker, whether a member of Congress, the Ag Committee, a CEO of a company, has a question of what is the state of knowledge regarding any question around the food system, we will do that evidence synthesis for them, combine the data, set the evidentiary standards, and give them the answer in terms of the health effects, the environmental effects, and the economic effects. And so we're building this in Fort Worth. We haven't even announced yet, and we have over $3 million in contracts already from just the beginning of the year. It's the only one in the world. You're going to see other people begin to copy already. But this is going to be really important in moving agriculture from really driven by preferences, values, beliefs, to something that is science-driven. So this is the Institute in the way that we have designed this. And we think of ourselves as an Uber or as an Airbnb. That is, we don't want a lot of infrastructure. We want to use our resources to address big problems focused on technology that are going to transform a particular area in the food system. And we bring in the very best people with the resources we have to address those problems. So that's the approach we use. And we created 3 hubs. One, of course, is precision nutrition. That's fundamental. If you're going to have health is the end point of the food system and dietary guidance, you need precision and we need to figure out how to do this. So we are working on technologies that we hope will do for nutrition what things like the Oura Ring and the Apple Watch have done for exercise science. And so we're working with those technologies, and we have collaborations all over the world with Intel on that. We have over at this end what we're calling responsive agriculture. And I'll define that in a minute. But agriculture has always been responsive to the endpoint. When hunger was the endpoint, agriculture responded and dealt with that. Well, how is the food system going to be responsive to human health and do it in a way that's sustainable both environmentally and economically? And we have a number of initiatives around there. I'll speak to one of them. But both of them feed into what we call Healthy Living Hub, and that is how do we do the important translational research in communities to do this work? Controlled feeding trials aren't helpful. They're good to understand scientific questions. But this work has to be done in free living people, where people need agency, make choices, and have the knowledge they need to enact what is precision nutrition. So we intentionally put these two as a feeder. This is the food environment. This is an individual and what they need in terms of their health. Both feed into what we call our Healthy Living Hub. We have 7 mobile laboratories, much like the CDC laboratories, but it's the next generation of the CDC. They give us their blueprints of their mobile vehicles. We can go anywhere in Texas with a fully equipped laboratories, freezers, all sorts of assessment to do studies that link responsive agriculture and precision nutrition. So I want to talk about one initiative we're doing in responsive agriculture, and hopefully some of you can engage with us. So as I mentioned earlier, you hear people these expectations of the food system. Human health. Some people focus on environment, right? We need to lower the environmental footprint. We all need to eat crickets or seaweeds or things like this. But people aren't going to do that, right? They're not going to do that. And the whole economic problem. Well, right now we have an economic system that is based on hunger. How do we move that to health? And how do we do it in a way that's going to be economically sustainable, so that agriculture is sustainable, so that young people want to go into agriculture, be farmers, et cetera, et cetera. So we have defined responsive agriculture in a particular way. We fundamentally say that yes, the outcomes are the environment, they are health, they are economics, but they're not all equal. Human health has to come first, because if food isn't about health, you're not going to get public support for it. That's the purpose of food. It's a major purpose is to promote health. So what is an agriculture system that supports health? And once we define what that means, how do we do that in a way that is economically and environmentally sustainable? We're not calling them equal. We're saying human health, figure that out, and then how do we go about adapting everything else? We're US focused. We're Texas focused. So we recognize the food system is global, this and that, but we're really focused on the US food system. So we started out and had kickoff with the partnership with the Chicago Council for Global Affairs. And in November, we had the Undersecretary in the USDA join us, and she's right, where is she? Where's Shavonda? Right there. But we had people from the House Ag Committee there. We had a lot of the leaders in ag policy and leading producer groups, leading public health groups, and began to roll out this idea of how do we achieve human health through responsive agriculture. Because again, health is about individual agency, individual biology, but it's also about the food environment that surrounds us. And so this kicked off this event, which now is leading to the next phase, which we're going to roll out probably within 2 weeks. We are going to continue our partnership with the Chicago Council on a 2-year project. And that is, we are bringing together leading experts in these 3 bins. We are going to have a committee on reducing diet-related chronic disease and really ask the question, what are the priorities we need? Both things we can do now, immediate, short-term, long-term, changes we need, or information we need, research we need, to make agriculture the solution to diet-related chronic disease. We know there are things we can do now. And then what are the barriers? Are they regulatory barriers? Are they a lack of will? Is it resources? What are the barriers? And so we're going to be putting out a call soon for experts to join this committee. They're going to be writing white papers on this, and they're going to be feeding into what's not on here is a task force that's going to integrate and communicate findings across these groups. This group is going to talk to this group here about, well, how do we then create a food system that then is going to support human health and do it in a way that, again, is both environmentally and economically viable, considering urban, suburban, considering technologies. What do we need to do to modify the food system to achieve this goal? And then a third one on securing nutrition equity. Again, most of the diet-related chronic disease is in the underserved population. How do we ensure that the information that we get from these first two bids then is actually going to benefit those most in need, those most at risk, those where you see most of the healthcare costs? So this is just our little schematic here. Top of the pyramid is chronic disease. This then feeds back to these two committees. And so we're really excited about this. It's getting very good support from Capitol Hill in terms of the ag committees, what they want to see done. And so we'll be doing a lot of work in Washington in terms of moving this information through as fast as we can. But our goal is to move the Farm Bill, if you will, or agriculture. That includes the nutrition title, includes all the farm titles, from being really politically divisive, where it is now, to being science-based and apolitical. We have a really good group, bipartisan group, who's supporting us and helping us do that. And again, we've done it before. The Green Revolution was all about using technologies to end hunger. So we have done it. We just need to do it again with what that new endpoint is. And that new endpoint is human health, and we need to have the economic and the environmental models that are going to support that. So with that, the research we do here, all of the research I showed you is Kendra Tiani's. She just defended her thesis 2 days ago at Cornell, so I drove down yesterday. She's a great graduate student, but she did all that mouse work I told you about. This is my research group. And at IHA, I am just so fortunate to work with two of the most brilliant people I know. Reagan Bailey runs our precision nutrition program. We recruited her to A&M from Purdue a year ago. And Rebecca Segan runs our Healthy Living Hub. She is the world's best at taking any sort of a health program, whether it's an extension program or whatever, and turning it into a randomized controlled trial that's participatory within the community, that's sustainable and everything else. She's absolutely fantastic. So the three of us lead the institute together. I'm nominally the director, but in fact, I just take orders from those two. And we value our collaboration with the Chicago Council. So with that, I thank you. I hope that was somewhat coherent. But that's sort of the approach that we are taking to deal with this big challenge of how we make agriculture and nutrition a solution to human health.
So we have this plaque to present. It says the Hans Fisher Lectureship, Enhancing the Purpose of Food, given to Patrick J. Stover, Ph.D. Thank you very much.
Thank you.
All right. Yes, big round of applause. Thank you so much.
It's always a pleasure to be here. I know so many of the faculty, and it's just a great, great place. So thank you.
We're very appreciative for your coming to join us. And that's it for today. Have a good rest of the day and a good weekend, everybody. Thank you all for coming.
Thank you all.